I'm pleased to be joined today by Dr. Michael Levin. He's a distinguished professor of biology at Tufts University, where he researches a wide array of topics ranging from learning and memory to regrowing limbs in animals. Mike, welcome to the Nature and Nurture podcast.
Thank you so much. Pleasure to be here. Thank you.
So, what is the central theme that connects all of your research? Because at first glance, these things, these cognitive things, and then looking at like limb regeneration and biomechanics are very different.
Yeah, so we do a lot of things in our lab, including some work in AI and cancer and synthetic living organisms.
Even more breadth than I alluded to. Yeah.
Yeah, many things. But there is this central theme that ties it all together. So these are all facets of one question, really. So what I'm interested in is embodied mind. I'm interested in how various diverse, and I'm talking about highly diverse substrates can put forth some degree of intelligence, competency, cognition, and so on. So we're interested in mind embodied in very different architectures. So we work in slime molds and cells and tissues and organs and whole colonies, for example, ant colonies and things like that. And yeah,
these are all things you might not ordinarily consider having minds, at least the average person who's not doing this type of research.
Yeah, and so if one takes the position that some things have minds and other things don't have minds, then one has to really specify what the binary criteria are. So I don't believe in a binary criterion. I have been working on a system of and certainly I'm not the first, a system of a continuum of agency, and the idea is to be able to specify what kind of problem spaces different systems work in and what competencies they have to solve problems in those problem spaces. And this, once you think of it that way, it does a couple of things. It sort of dissolves a bunch of pseudo problems that people get hung up on all the time, but also it provides very specific research agendas. So for example, in thinking about morphogenesis as a collective intelligence of cells trying to navigate morphospace, so the space of possible anatomical configurations, and what morphogenesis, let's say during embryonic development or during regeneration, what it does is it tries to navigate from one spot where it begins to a target morphology, a region of morphospace that is appropriate to the whatever shape it's trying to build. So looking at that as a kind of collective intelligence that has goals and various competencies to get there opens up a whole new research program, because then you are not limited to trying to manage morphogenesis bottom up by manipulating the hardware of the system, but you start to ask computational questions. What does it know? What does it measure? How does it know where it's going? Could we train it? Could we use the various tools of neuroscience to manipulate its competencies? Are there stress minimization? Is there active inference? All of these things, and these become research programs. So
the morphogenesis, that is the process by which cells decide what to become, like become a limb or a head or whatever.
Well, to be specific, it's first of all, it's not just individual cells; it's collectives. So no individual cell knows how many fingers you have or what a finger is, but the collective certainly does. And it's not only the initial step, which is making decisions of how to become specific shapes, for different organs, the whole body plan, but actually the decisions it makes after that to defend that shape against injuries, accidents, aging, cancer, and so on. So there are many examples of that.
Even if there aren't any binaries here, I imagine there's a massive switch when you go from like a single-celled organism to multicellular in terms of complexity and what happens there. So what does that shift look like?
Yeah, there are many ways to think about that shift, and so some people think about it as a shift of evolutionary individuality and various other things. One lens that I find useful is to look at shifts in terms of the intelligence, and what I mean by intelligence is a kind of William James's definition, which is the ability to reach the same goal by different means. So basically, problem-solving competencies. And so what happens in going from individual cells to multicellular structures is two things. One is a change in the problem space in which it works. So single cells solve problems in metabolic spaces, in transcriptional spaces, and so on. But collectives of cells can solve problems in anatomical morphospace, meaning that they can work together towards a goal that's making an organ or making a body structure. And of course, there are other spaces after that. So there's the three-dimensional space of conventional behavior. There are linguistic spaces and so on. And so one thing that happens is that the system becomes capable of having goals in other problem spaces, but also it enlarges what I call the cognitive light cone. So the cognitive light cone is basically a kind of diagram that tries to semi-quantitatively map out the size of goals that it can work towards. So if you think about, so all creatures, including evolved, natural synthetic beings, all kinds of creatures, one of the things that ties them all together is having a particular shape of a cognitive light cone, which is what is the size, literally the spatiotemporal size of the goals that it can work towards. And so the thing that happens during multicellularity and then collapses during disorders such as cancer is this inflation of the cognitive light cone. The goals that it can produce become much bigger and more complex.
When people talk about the goals of an organism, especially from an evolutionary perspective, some take it very literally, like the organism always has its own drive to survive and reproduce. And others treat it as this sort of passive process, like goal is just a metaphor for the process that evolution naturally unfolds. How much intentionality do you attribute to animals across this complexity spectrum?
Yeah, well, let's dissect that in a couple of ways. So first of all, I take not only individual animals as having goals, but lots of their components do. So your various organs have goals, your individual cells have goals, the molecular networks inside those cells have specific goals in various spaces. We as humans are terrible at recognizing intelligent goal-directed behavior in unconventional spaces. We're very good at noticing medium-sized objects moving at medium speeds and achieving goals in three-dimensional behavioral space. But if you, for example, had an inborn, so kind of an innate sense of your blood chemistry, you would be very quick to be able to recognize your liver and your kidneys and so on as solving really interesting problems in physiological space. And so we need to be really humble about being able to detect these things. I take, how seriously do we take these metaphors? I mean, the first thing I want to say is that all science is the construction of metaphor. We have nothing. So people will say, wow, that's just a metaphor. That's all we have. There's nothing else. Everything is a metaphor. And so if you want to talk about human goals, of course, there are people who will say that, well, humans don't have goals either. They just have, they're complex kinds of systems that respond to specific stimuli, and then whatever happens. So I take goal talk very seriously. I think that goal directedness is an incredibly powerful metaphor because it helps you make predictions, it helps you do new science, and it helps you reach new capabilities, which means that in order to make claims about goals, you can't just sort of sit back in your armchair and say, thermostats don't have goals, I have goals. These are, you can't make these philosophical pronouncements, you have to do experiments. So what that means is you have to specify a problem space, you have to specify what you think the goal is, and then you have to make a hypothesis about how much competency does the system have to reach that goal, and then you do an experiment, and then we find out if your set of hypotheses was useful or not. And so these are all empirical questions. And for us in making goal-centered models of regeneration, of development, of various things like that, we've reached lots of new capabilities that no one had done before, and that new discoveries, new research programs. So I take that to be exactly what metaphors in science are supposed to do. So I take it very seriously.
When you're developing a plan of research, how do you decide what animal to use, ranging from like a very small planarian up to a frog or whatever other level of complexity?
Yeah, I mean, we go way broader than that in that we not only go down well past planaria into cells and bacteria, but we also think about synthetic forms, in silico forms. There's a very wide range of possible model systems in which to study this stuff. I mean, and the criteria are, you want to balance, you want an example that you want something that really illustrates very well the phenomenon you want to study, but you also need it to be tractable. So it needs to be something that current or adjacent possible tools will allow you to really make a dent in the understanding thereof. So that's so all these different model systems are good for different things. In silico models are really good because you control exactly which aspects you want to include and which aspects you want to ignore. So that's very useful. And also they're very fast. And so you can do, millions of generations in a simulation that you couldn't do in real life. Planaria are very good to understand natural mechanisms of regeneration, for example. But one thing you can't do in planaria is practice new regenerative therapies because they're already so good at it. There's nothing to improve. They're perfect regenerators. So for that, we usually start with something like frog, which has limited regenerative capacity. It has tissue renewal the way that humans do. But as an adult, it has very limited regenerative capacity. So we try to improve that. And then for human relevance, you might go to human cells and culture, or you might go to organoids or mouse or something like that. So, you want a tractable model of whatever you're studying.
Where did your research start and how did it evolve?
Wow. Yeah, I guess that depends how far back you want to go. So
We can go back as far as you like.
Well, I mean, I've been interested in this stuff since I was a kid. So very young, I was really interested in engineering and the understanding of electronic artifacts like televisions and how it was that, I mean, we're talking, I don't know, 5, 6, 7 years old. I would look into the back of a television set and just be just amazed that somebody knew how to put all those parts together to make it work the correct way. But at the same time, I would go outside and look at beetles and caterpillars and various insects and think about what the commonalities and differences were. So these things also made of parts. How did they get here? How is it that they seem to have preferences and behavioral repertoires that our machinery doesn't have? And what's the commonality there?
You think there's something about relative simplicity, like I'm assuming you're talking about like an old school boxy television, which you could actually take apart and maybe get a sense at how it's working. And nowadays, like you just have a very compact screen, you can't take apart at all. And I imagine in biology, it's somewhat similar, right? Like you might think that the smaller the animal, the simpler it is, but then also it's like once as you can look at a human and then kind of just at a more macro scale, look at the limbs and what they're thinking and saying and kind of get a broad idea of what's going on, even if you know nothing about the cellular biology. But then the smaller you go, it seems like the more you need that cellular biology knowledge in order to get a sense of what's going on. Do you see a parallel there?
Yeah, I mean, I think it's all in the eye of the observer and what kind of questions you're asking. I mean, I don't think there really is any simplicity in biology anywhere. I think we impose certain kinds of simplicity when we as scientists make decisions about what we're going to ignore and what we're going to try to focus on, which is essential, right? The whole point of science is to be able to say something without having to say everything. And when people say simple organisms, I mean, look, yes, you're not going to study complex psychological drives and social behavior, some societies in planaria. But so of course there are ways in which they're simpler, but all of the real mysteries of what it means to build a body, to repair a body, to have a self that's more than the sum of its individual cells, all of that was solved long before planaria came on the scene. So
Mm-hmm.
for the wide range of problems that we're really interested in, planaria are no less complex than any other organism. I mean, they're incredibly complex. If we understood planaria, we would be done with a lot of science, certainly a lot of the biomedicine that we need. I don't consider them simple.
We
Simple at all.
should introduce to our audience what planaria are, and then we can return to your story.
No, sure. Yeah, planaria are a kind of a free-living flatworm. They have a brain, they have various organs, they are similar to our ancestors, so they have true bilateral symmetry. They are amazing in many different ways. One of them being that they can be cut into pieces. I think the record is something like 275 pieces. Every piece will regenerate a perfect little worm. So every piece knows exactly what a worm looks like and will rebuild all the missing structures. They seem to be immortal. There doesn't appear to be anything like an old planarian. They sort of don't age at the individual level.
Wow.
They're smart, so you can train them. And if you do train them for specific memories, and then cut off their head, including their brain, the tail will sit there for 8 or 9 days doing nothing. They will regrow a new brain and then show evidence that they still remember the original information. So they have this kind of distributed memory storage and the ability to imprint those memories back onto the newly developing brain. They have various other interesting features. They're very cancer resistant, but if you do induce a tumor and then you trigger regeneration elsewhere in the body, the tumor tends to get cleared up. So they have this amazing sort of remodeling kind of capacity. Planaria that are starved will simply shrink allometrically, so they shrink in perfect proportion, and then you feed them and then they grow in perfect proportion. And then the final thing, which bears in interesting ways on this whole question of genomes and what genomes do, they have an incredibly messy genome because they tend to reproduce by cutting themselves by splitting in half and then regenerating. So this means that they accumulate somatic mutations, and they can be mixoploid. So every cell could have a different number of chromosomes. They're a total mess genetically, and yet they're champion regenerators, with really excellent solid anatomical control. So that's kind of an interesting puzzle that we can talk about.
Is there more variety in their genome compared to an organism of similar size?
What do you mean by variety?
In terms of the mess you were just talking about.
Oh yeah. I mean, they could. You're right. Yeah, they could. Like I said, they could be mixoploid. So cells could have different numbers of chromosomes, all in the same animal.
And that's not typical.
No, it's typical of tumors, but it's definitely not typical for normal cells in your organism. They all have the same. Almost all of them have the same genetic complement.
Interesting. Okay, we should return to this topic, but I want to let you finish your starting in your childhood and then talking about how you first became interested in these topics.
Yeah, so I was interested in this issue of life and engineering, and I thought that primarily I was interested in mind and these questions of where do things like, I mean, clearly we're all made of some kind of parts, but somehow we acquire preferences and goals and memories and various cognitive capacities and a first person inner perspective and things like that. So I thought that I was a coder from an early age. I programmed, and I thought that really the way to understand these things is to try to build new ones from scratch. So artificial life, artificial intelligence. And so I went as an undergrad. I went for a computer science degree. I then sort of halfway through realized that we just didn't know enough to do this without the biology, and so I also got a biology undergrad degree. And then from there, went to grad school. And at that point, I was studying left-right asymmetry during embryogenesis, how the different organs figure out which side is left and which side is right. And that's a very interesting problem because it gets to this kind of collective decision making. So to figure out whether you're on the right or the left side of a midline is a collective decision problem that requires some kind of emergent whole to exist. So there's got to be a midline, and there's got to be this large scale geometric kind of information that cells need to make decisions based on. And so I worked on that, and then as a postdoc, I sort of pushed that backwards from the genetics of it to the biophysics of it, because of course genes can't tell left from right. So there's a biophysical phenomenon at the root of it. And so I studied that, and then in 2000 I started my own lab. So this was at the Forsyth Institute on the Harvard Medical School campus, and at that point we started. We had made some of the first molecular tools to read and write the electrical information that the groups of cells are using to store pattern memories. And so this is developmental bioelectricity, which of course people had been working on for a really long time, but we had made some of the first molecular tools. And so at that point, my lab began studying and developing these methods to literally read and write the cognitive medium of cells to look at a set of tissues and say, okay, what is the information that they're processing? It's very parallel to what neuroscientists are doing in the brain, but this actually happens in every tissue in your body.
Mhm.
So we studied, and so we started applying that to different scenarios. So regeneration, birth defects, cancer. And so then I moved to Tufts in 2009, moved the whole group over to Tufts in 2009. And then we continue all of that work and then added a bunch of stuff using computational models, synthetic biology. So create a synthetic morphology. So creating novel living organisms. And that's kind of the evolution of it. But basically, working up from the molecular and biophysical mechanisms of cell communication to really try to understand first the computations and then the cognitive aspects of those computations to understand decision making in these other spaces.
Was it gradually working your way down in realizing, let's say, the complexity of intelligence that first maybe starting in artificial intelligence, if you had this computer science background, and then realizing we need to understand the biology to understand intelligence, and then perhaps going towards animals and realizing we need to understand cells to understand animals, and then we need to understand the molecular biology to understand the cells? Is that what happened?
No, it's actually the other way around. It's this continue working upwards, actually, because I mean, it's very obvious to everyone that understanding molecular networks and cells and tissues is important, but the vast majority of people, I think, to this day, see it in a very bottom-up fashion. In other words, that if we, that the best way to interact with these things is in a kind of a reductionist approach where we focus on the hardware. So look at all of molecular medicine, right? It's all focused on the hardware. It's a single molecule kinds of approaches, genome editing, CRISPR. This is all hardware. And with, from my computer science background, I always thought that, the hardware is great, but look at how much power you get if you are able to understand the software and program and take advantage of the reprogrammability of these things at the software level. And so working up from the molecular mechanisms of what's going underneath to then ask bigger questions is, yeah, but what are the computations? What's the system? What's the information processing that the system is doing? And then working up from that and saying beyond computation, there are cognitive aspects here. So what does the system know? What does it expect? What does it remember? What does it measure? And how do we communicate with it? And how do we relate to it? So sort of working upwards from the physical details of the machine to the computations to finally the sort of aspects of intelligence and cognition that I'm really interested in.
Could you describe what the computational research that you're doing is and how that complements the biology?
Yeah. So we have a variety of computational approaches. One set is to construct relatively biorealistic models of cellular signaling to really look at mechanisms by which, for example, a whole tissue can decide whether the pattern is correct. Right, so a planarian or a salamander is able to regenerate missing structures, and so for that you need to understand how do cells know whether they've hit the correct pattern or not. When do they stop? What should they build? So making computational models of the bioelectric communication in cellular networks using some tools of connectionist to machine learning and other kinds of paradigms to try to understand how do collectives make decisions in morphospace. How do they navigate? And so these are computational models of morphogenesis. And those
Is that
models,
where cancer comes in? Where if the signal that you're done doesn't kick in properly, then the cells just go out of control and keep multiplying?
no, cancer is, well, it's related to that, but cancer is a slightly different story. So I think that what happens in cancer is a reduction of the cognitive light cone. So basically what that means is when you have a collection of cells and they're all in very particular electrical communication with each other as a network, that network can store large-scale pattern memories and work towards implementing a heart or an eye or a liver or whatever it's making. Individual cells that become disconnected from that electrical network they're not any more selfish than the other cells. It's just that their cells are smaller. They're tiny. They are no longer connected electrically to this other network. And so all the goals that they're able to pursue are single cell scale goals. And what do individual cells want? Every cell wants to become 2 cells, and they want to go where life is good. And that's metastasis and overproliferation of cancer. So in cancer, that large cognitive light cone shrinks down because of the breakdown of the electrical communication. And then the cells basically revert back to their ancient unicellular lifestyle. That border between self and external environment, the individual cancer cells basically see the rest of the body as just external environment. They're just amoebas living in a wide world at that point. And so of course they're behaving in ways that are maladaptive for the rest of the organism. Yeah.
What is the incentive for the organism to like narrow and specialize the cells, as opposed to throughout the entire lifespan having everything be a stem cell that has that flexibility to become whatever it needs?
Well, so there's a couple of things. I mean, becoming everything it needs. Let's remember what stem cells are able to do. So individual stem cells can become other cell types, but that's not really enough. So your hand and your foot have exactly the same cell types, but so they have bone and skin and muscle and whatever the other tissues are, but they're different. So having stem cells become individual cell types is not sufficient. It's necessary, but it's not sufficient.
So that's like a hardware software question. The hardware is the same, but they're operating on different software.
Basically, I mean, in general, what happens is that the genome provides the cellular hardware. So the genome tells every cell what tiny components it gets to have, so all the proteins. But then after that, cells need to interact and form networks in ways that make decisions at the physiological level. So those decisions, right from the beginning, when a bunch of embryonic blastomeres get together and become an embryo, what's an embryo, right? So it's a set of cells that are pursuing specific paths through anatomical morphospace and together as a collective. And so specializing, and again, this isn't exactly my field, but people who study the evolution of specialization, we have many stories about why it pays to have cells with specific functions in your body, right? It's it's a highly adaptive architecture.
You mentioned embryonic development. That's another topic that typically gets this sort of binary, maybe false binary, placed on it. Like, at what point does the embryo become human or become whatever organism it's becoming? I'm guessing you see it as more of a natural continuum. Could you shed some light on that?
Yeah. I mean, these issues of when does something become are universally pernicious, meaning that they provide no value other than no value at all, and they generate all kinds of pseudo problems, because trying to find a bright line at which on day X of development you are now conscious, or you are now a human, or you are now a cognitive whatever. There's never been a good story about that ever, right? So people make it up. People will make arbitrary lines, and and if for specific reasons of medical ethics and whatever, sometimes you pick a line just the way that it's a little bit like when I talk to my students, it has the same status as the word adult, right? So in society we have this notion of an adult, because and at 18 you become an adult, and then various other things will happen in court and so on. But we all know nothing special happens on your 18th birthday. It's a total fiction. It's a complete fiction that's designed to lubricate the practicalities of social life. The same thing here. You can draw lines for various regulatory issues if you want, but they don't exist. There's never been any convincing sharp mechanism where boom, you go from here to there. I mean, just think about what an incredibly slow and gradual process development is. There's stuff happening all the time. It's impossible to pick any one special time frame where something discrete happens. And the same thing with evolution. When people say the human, I guess they typically mean a modern adult human. But if you sort of walk it back, right, generation by generation, you get to our various hominid ancestors, and then very slowly to various other things that are kind of lower primates and so on. And there's just no sharp line. If you wanted to say, well, humans have I don't know, pick something, true responsibility for their actions, and animals don't. I mean, that binary category completely dissolves when you take evolution seriously. You sort of walk it back and you say, Well, you're telling me that at some point there were some hominid and there were some mammals that didn't have it, and then these parents, boom, they had an offspring, and now it has it. I mean, that's
Mhm.
it's just completely implausible.
So
So yeah,
here's something that might have a sharper line. At what point do you call something living?
yeah, that may have a sharper line. There are people who study that question. So people like Lee Cronin and Sarah Walker and folks like that. I don't know. I don't work on that, and frankly, I don't find the category living. I don't find that a particularly interesting. I mean, this is crazy for a biologist to say. I know that, but I don't find that a particularly important distinction.
And I
So
don't mean just across development, but I mean like at an organism level, like
yeah,
at what level of complexity or
I mean, well, you know, certainly every part of development is living. So eggs are living. You know, the single
Mhm,
cell egg is living. The way people do think about this is in terms of the origin of life. So at
mhm,
one point you had an abiotic Earth with what they call just chemistry, and then eventually you have life. So
mhm.
okay, there are like the folks I mentioned who work on specific criteria for what it means for a planet to have life on it. And that's fine. I don't work in that field. I'm much more interested. I'm less interested in the Venn diagram of things of living and non-living, and much more interested in cognitive. So I think that
Mhm.
there are many systems that are cognitive but non-living. There, at this point, there are probably no systems that are living and non-cognitive, but maybe there were at the beginning of life. But yeah, I'm much more interested in the spectrum of cognition. I'm not sure what to say about whether life is continuous or not.
What does cognitive but non-living look like?
So let's take a step back and just think about what I mean by cognitive. So when I say cognitive, I mean the spectrum of diverse competencies, including extremely primitive ones. I mean, at some point, anything has to have kind of a basement, the lower end. Uh, all of these competencies to solve specific problems. So there's a couple of ways to think about it. One way to think about it, and this is a great a line that I wish I could remember who said this, but someone once said, it's the difference between two magnets trying to get together and Romeo and Juliet trying to get together. So what's the difference? The difference is in what degree of competency do you expect in that process. So the two magnets are basically just minimizing their distance. If there's a barrier, that they're going to stand there, pressed up against the barrier. That's the end. You're not going to get an awful lot of. It's not zero competency. They can do something, but it's awfully low. Whereas from Romeo and
Mhm.
Juliet, you can expect very sophisticated kinds of behavioral actions in their problem space to try and get their goals accomplished, and then everything in between. Right? We have all kinds of systems in between.
Is
So
this the same as that light cone metaphor?
it's related to that, right? So the light cone measures the size of goals you can achieve. So let's just talk about that for a second. If you tell me what the largest goal that you care about is, I can basically guess your intellectual sophistication. So if you tell me that all you care about is the local sugar concentration, you're probably a bacterium, right? If you tell me that you're capable of caring about what happens in the next a couple of days, but you really can't. You really don't have the cognitive wherewithal to care about what happens 3 months from now in the next town over. You might be a dog. You might be something at that level, right? Where you have a degree of concern over a spatiotemporal area that has a particular radius, but let's face it, you're never going to care about what's going to happen next year. It's just not possible for you to care about what happens next year, let's say. If you tell me that your goal is world peace or something, or the stability of the stock markets across the earth over the next 100 years, I'm going to say you're at least a human. And if you tell me that you are capable of caring in the linear range about thousands, millions, or trillions of living beings across the planet, I'm going to say you are something that's beyond current humans, because current humans can't do that. They don't have the cognitive capacity to care in the linear range about that
Uh,
many other beings.
when you say non-linear, does that mean it's like one life is a tragedy, 1,000 is a statistic? Like we
Yeah,
start to just clump things together?
yeah, basically, right? So human beings simply do the standard modern human beings simply do not have the cognitive capacity to exert to multiply the amount of care that they would be able to muster for one person or a small number of people over to a large. Just as an example.
Mm-hmm.
So the cognitive light cone measures the size of your goals. The what I call the spectrum of persuadability measures the sophistication with which you are able to pursue those goals. So you can imagine the spectrum where on the left are extremely simple things like mechanical clocks. In order to change what they do, so in order to change what that does, you have to rewire the hardware. You're not going to train it. You're not going to convince it. There's nothing to do except rewire the hardware. So you have things like that. Then you have some slightly more interesting things like thermostats. So thermostats are cool because they have a set point and they try to maintain that set point. And in order to change what they do, you don't actually need to rewire the hardware. You can leave the hardware in place, but you can change the set point. And then you may have slightly more sophisticated beings that, yeah, they have a set point, but they also resist it when you mess with their set point. So they have this kind of like very weak metacognitive capacity where they know when somebody's trying to change the set point and they fight you on it. And so then of course, this is a very dense spectrum, but there are many, another interesting position is creatures that can be trained with rewards and punishments. So you can't do that to a thermostat, but there are lots of other kind of reinforcement learning types of agents and living things that you can train into new behaviors with rewards and punishments. And then further to the right on that spectrum are other beings whose behavior you can change with communication, with messages, with rational reasons as opposed to causes. And so this whole spectrum. And so you need to different beings land in different places on this spectrum. And so to go back to your original question, what does cognitive but non-living look like? I can easily imagine, in fact, we already have engineered creatures that you wouldn't say are living with this. And this is why I'm not particularly interested in living as a distinction. There are lots of creatures that you probably wouldn't say are living, which are somewhere on this, occupy some interesting place on this continuum. So they can either learn, they have goal-directed activity, maybe they have some open-ended ability to set their own goals. I mean, we're still sort of working up to that in AI, but yeah, there's nothing. I don't believe there's anything magical about the protoplasm. There's nothing magical about the stuff that we're made of. And no doubt in the wide universe, there are other cognitive beings that don't look anything like us. Whether you want to call them living or not, I don't know. But cognition is, I think, what we ought to be tracking, not the stuff that we're made of or how we got here.
This ability to learn is a great bridge back to planaria. Perhaps we could start talking about some of the history of how learning and memory has been studied in them, and then move up to your more recent work.
Yeah, well, people have been studying behavior in planaria for well over 100 years. A particular milestone was in the 1960s, the late 60s, when this guy James McConnell discovered that you can train planaria and then cut off their heads, and they will regenerate new heads and then show that they now remember the information. So McConnell did some amazing work on this. He caught a lot of flack over it. Lots of people wouldn't believe that that's true. We reproduced in 2013. We made an automated device. My lab made an automated device that took all the human element and the guessing out of it and proved that he was absolutely correct, that they do in fact do that. And there's a tremendous research program going forward that can be done to understand where is the memory, how is it stored, how is it imprinted onto the newly developing brain. It's a fascinating. Now it's related to work that was done in the 70s by a bunch of Russian scientists, and then more work was done later by Douglas Blackiston, who actually is a staff scientist here in my center, who discovered that through the caterpillar to butterfly or moth transition, when the brain is basically taken apart and reassembled in a new way, again, information persists. So training persists. And so that's just, amazingly interesting. It also raises all kinds of interesting sort of cognitive philosophical implications, where we often in philosophy 101, they often tell you to, imagine what's it like to be a butterfly, or what's it like to be a caterpillar? Well, that's a basic question, but the more interesting question is, what's it like to be a caterpillar changing into a butterfly in real time, right? So you are actually, so it's not just an evolutionary thing, it's not just a developmental thing, but you are actually an animal whose brain and whose cognitive capacity is totally rewired and changes radically, right? You used to really care about leaves and eating leaves and what kind of leaf, and now you don't care about leaves at all. You want nectar, and now you live in a three-dimensional world instead of the two-dimensional world of a caterpillar.
Mm-hmm.
It's an amazing journey, you know.
And that seems like a radically more extreme version of the question, like what makes you the same person over time if all of your cells die and are reborn?
Yep. Yeah, that's exactly right. And so some people are interested in memory as a marker of continuity. So if you share the same memories, then you are the same person, right? And I mean, there's a more basic version of it that all of us went through. So we were all children at one point, and then you sort of go through puberty and lots of things change, right? Your body changes, your brain changes, there are new hormones. The things you care about change radically. And your capacity to have goals and various other psychological drives change radically. In what way are you the same person? So I think the closest that anybody can formulate that is well, you have memories, right? There's a memory identity, but that's actually tricky, because one thing you can do in planaria is you can cut them into pieces, and a single trained worm with a certain memories can give rise to 10 other worms that all have exactly the same memories. And so now, where is the individual identity? And so I think, much like with everything else, this binary question of are you or are you not the same individual, I think that question leads us astray. I don't think that's the right question.
So at some point you lose that ability, or maybe planarians are unique in having this ability to regenerate. But that seems like it would be the most adaptive thing you could have, to be basically immortal and able to regenerate yourself. So is it too simple a question to ask why can't other organisms do that?
No, it's a very good question. Some organisms do it to an extent. So salamanders regenerate their limbs, their eyes, their jaws, their spinal cords, portions of their heart and their brain. They're not quite like planaria, but very, very regenerative. And mammals regenerate a little bit. So we regenerate our liver, deer regenerate their antlers. So it really is a very good question as to why doesn't everything have this amazing regenerative ability? Why are we not like planaria? So I don't know the answer to that. I don't think anybody knows the answer to that, but I could try to tell a story that may or may not be true. So imagine that you are an early mammal. You know, you're kind of a mouse-like creature. You're running around the forest. Something bites your leg off. Now, unlike a salamander, a couple of things are different. Number one is you don't have buoyancy, so you're going to try to put your weight on it, which means that whatever delicate cells are trying to regenerate at the wound, you're going to immediately grind them into the forest floor. So they're going to get dirty, they're going to get infected. You are much better off sealing that wound, scarring, and hoping you survive, rather than leaving it open and trying to regenerate. Another issue is that because you're not aquatic, so with only one exception that I know of, all good regenerators are aquatic. And so without the water on the other side of the wound, you really can't run injury currents very well. So all the bioelectrical signals that are normally important to regenerate, you can't run them in dry air. And so that's another reason why that might be very hard. So I think what's happened in mammals in particular is that the evolution has sort of gone in favor of let's seal the wound and do our best that way. But then, of course, there are also species of planaria that don't regenerate very well, and there's a wide variety of different animals that can and can't regenerate. It's unclear.
Why do you think this doesn't apply to brains? I mean, we have neuroplasticity, but in terms of, like, if you get a brain lesion, you probably won't regrow it that area, or regain the function.
Well, I don't know if it's anything special about brains. The same thing is true of your finger. If you lose your finger, you're not going to regrow your finger. But planaria regrow their brains.
Right. Because with limbs, we have this thing you just talked about, where it can get damaged or infected, so maybe it never could fully regrow. But you would think that your brain is protected within the confines of your skull, so maybe that's like a safe haven to regrow.
Yeah, it's a good question. I don't know. It's possible that this shift to scarring in away from regeneration is just global. you mean, I told a story about limbs, but maybe it's hard to do that in specific regions and not others. And so maybe just everything is like that now in the human body. But like I said, salamanders and planaria regrow, well, planaria regrow their whole brain. Salamanders can regrow portions of their brain. I don't think there's anything specific about the brain that forbids regrowth, but there may also be trade-offs, evolutionary trade-offs that we don't know. Also, it may be that this is good enough. Remember that evolution doesn't optimize for your happiness or for a high quality of life or anything like that. It optimizes for biomass, so it's good enough. If you can survive this way, then that's a valid solution. I apologize. Give me 30 seconds. I'll be right back, and hopefully somebody's ringing my doorbell. Hopefully you can.
And we're back. So Mike, with our last 10 to 15 minutes, let's talk about the future of your research and where you see the field of regenerative medicine and synthetic biology going.
Yeah. So there's a few things. One of the things we hope to contribute to is regenerative medicine broadly. So the idea is, so we work on some specific things like limb regeneration and birth defect repair and cancer reprogramming. I think the future of the field is really going to parallel what happened in computer science. So when I give talks, I often show this picture of what programming looked like in the 40s and 50s, which is, you see this person sitting in front of this giant kind of electronic thing, and she's plugging the wires in and out, right? So you were programming at the hardware level. But one of the most amazing aspects of computer science that led to this information technology revolution is this idea that if your hardware is good enough, and I think biological hardware is definitely good enough, you gain this aspect of reprogrammability, meaning that you can communicate with the device via inputs, via stimuli, not physical rewiring. And I think this is the future of regenerative medicine. So the future of medicine, future medical treatments are going to look more like, we've used this phrase in some of our writing. It's going to look like somatic psychiatry. So the idea is not going to be trying to rewire the hardware at the molecular level. It's going to be what stimuli and experiences can you give this collection of cells to convince it to do A or B. So whether in morphological disorders, whether in physiological disorders, it's going to be more about communicating with the collective intelligence of the cells and try to nudge it into particular regions of the space in which it works, and as much as possible taking advantage of its native competencies. So you don't want micromanagement. You want to be resetting goals and taking advantage of its innate intelligence. So I think that's going to be so tissue training regimes and things like that, I think, are the future of regenerative medicine.
So is gene editing analogous to the hardware editing, and then this is something more like manipulating epigenetics?
I'll agree with the first part, which is that, yeah, genome editing changes the hardware. So what the genome does is it tells every cell what molecular hardware it gets to have. So it's sort of the equivalent of saying, this particular type of junction between elements, we're going to change its properties. So you can imagine how hard it is to program that way, right? And so to control a complex computational device that way. So I think certain low-hanging fruit, single gene diseases and some things like that will be solved by genome editing, but that's it. After that, it's going to very quickly run into a brick wall when it's completely unclear which genes need to be edited and how to make large-scale anatomical changes. The epigenetics, I think people have a lot. People put a lot on epigenetics than I think is warranted. So traditional, the modern understanding of epigenetics is things like chromatin modification. So functionally, all that is another way to turn genes on and off. So there's no more magic to it than that. So in the end, it's still that aspect of it. Chromatin modification is still single cell level kinds of phenotypes by turning genes on and off. So I think that's very limited. There's certain great things you can do with that, but it's not a broad answer to these things. On the other hand, the original meaning of epigenetics was anything that's not genetic. And so from that perspective, yeah, there are types of epigenetics, for example, bioelectrical kinds of circuits that are not genetic. but they're not what people typically mean when they say epigenetics. Typically, they mean, sort of chromatin remodeling and things like that. And that I don't think is radically different than the standard genetic approaches.
So is this going to be a bottom-up approach where by seeing how, like, let's say some sort of bioelectric stimulus changes a single type of cell in one organism, then you can kind of move up the complexity ladder and hope that it generalizes and then maybe up to humans eventually?
No, I think it's exactly the opposite direction, which is that the bioelectric code is not, these are not single cell properties. So the idea is that it's very much like the way electricity is used in, let's say, neural networks, whether artificial or biological, to store memories, to implement behavioral repertoires, and to implement intelligence. And so the idea is going to be to understand how all cells at whatever, whether human or not, that doesn't matter. These things are extremely highly conserved. So, and to understand how all cells implement intelligent, some degree of competency in goal-directed behavior in their electrical signaling, and learn to read and write those patterns to take advantage of it, to hack into the native competencies of the tissue to get either restoration of native patterns, so anatomical repair, or in fact make new patterns. So, biorobotics, synthetic living machines, those kinds of things. And it's not going to be done bottom up. I don't believe it's going to be done bottom up. I think that the much more interesting and powerful way to do it is top down, is to ask, how does this system natively represent information? How does it store its goals? How does it pursue those goals? And now what kind of stimuli how do we communicate with it, literally communicate with it, to re-specify those goals and take advantage of its native competencies?
So modern neural nets can do amazing things, but they're kind of a black box. Like, we don't know exactly how they program themselves to get to the outcome. Do you see the future of synthetic biology kind of similar to that, where maybe we can create very complex cognitive machines, but we don't know exactly how they're working?
Yeah, it's an interesting point. One of the, I forget which one, but one of the, maybe it was Bohr or somebody, one of the sort of fathers of modern physics had said that everything is understandable, but we can't make any assumptions on what understanding actually means. Meaning that for certain systems, understand might be not the same as, which it makes perfect sense to me that I'm not sure why we necessarily expect that the same cognitive tools that we use to understand simple systems in an everyday world, which is how our cognitive systems evolved, right? Why those same kind of conceptual tools would be suitable to understand every phenomenon in the universe, including, for example, intelligent behavior of networks. Doesn't seem like that's a requirement at all. And so it may well be that we have to have different notions of what it means to understand certain phenomena, especially highly emergent phenomena. The whole point of strong emergence is that you can understand the behavior of individual subunits in a way that there's a discrete kind of transition to a new kind of behavior. And when you say you understand it, what you really mean is that you understand the parts and you can specify what's going to happen and when, but that's it. And so I think it's entirely possible that that's where we end with that there is no understanding to be gained of these neural networks in the same way that we understand much simpler phenomena. But I don't know that that's true. I just think it's a perfectly good possibility that it may not be because of our ignorance. It may be fundamentally that they require a different notion of understanding. The same thing, I mean, that to some extent is true of biological networks too, meaning that we do not have a mature science of reading and writing information in and out of the brain. So if I were to look at your brain, I would have very, very limited information on what you know and what your goals are and all of that. But you know a lot of it. You don't know all of it. So internal access, privileged access, is not perfect by any means. We are often very much misled about our own abilities and goals and so on. But nevertheless, you know a lot about what you know, and internally, right? So there's some kind of interesting magic about this kind of system having internal metacognitive access that, looking at it from the outside, we still are not able to do. So I think this question of what can we really understand as outsiders without being the system itself, right? And so maybe the only way to gain perfect understanding of the cognitive capacities of a network is to be that agent. It is to be that network. It may not be possible to do it from the outside.
This has been incredibly stimulating, Mike.
Thank you so much. Yeah.
Thank you
great.
very much for your time.
Yeah, much appreciated. Thank you.